Enzyme is one of the most important compounds in living organisms. This is a very important topic in biochemistry. Enzyme is an organic substance that acts as catalyst in living organisms, regulating the biochemical reactions without itself being altered in the process. Hello everyone, welcome to my channel Dr. Tapati's presentation. Myself Dr. Tapati Bhanjade. Today I am going to start a lecture series on enzyme. This is the first video. For further notification, don't forget to press the bell icon. Kindly stay with me till end of this video. Let's start the presentation. Unit 1, Introduction to Enzyme. In this unit, I am going to give you a small introduction to enzyme. Then I am going to discuss about different properties of enzyme and effect of different factors on enzyme activity. Several chemical reactions are involved in all the biological processes or cell metabolisms. Various enzymes regulate all of those biochemical reactions. Enzymes are required for both catabolism as well as for anabolism process. Catabolism means breakdown of larger molecule into smaller molecules. As for example, digestion of food in which large nutrient molecules like proteins, carbohydrates and fats are broken down into smaller molecules. I hope you are familiar with pepsin, trypsin, amylase or lipase. Those are well known as digestive enzymes. Digestive enzymes are required for the digestion of food. Anabolism means synthesis of larger molecules from smaller molecules. It is just opposite to catabolism process. As for example, construction of cellular macromolecules like protein, carbohydrates and fat from their smaller precursors like amino acid, sugar and fatty acids. Hence, we can say that enzyme is one of the most important compounds of living organisms. Enzymes are present in all living organisms. They are mostly proteinaceous in nature. Enzyme converts substrates into products. It acts as biological catalyst. Therefore, it increases the rate of biochemical reaction and remains unchanged in chemical reaction. I am going to discuss all the properties of enzyme one by one. Most of the enzymes are three-dimensional globular proteins, except there are few RNAs which act as enzymes and they are called ribozymes. As for example, hammerhead ribozyme. Proteinaceous enzymes have tertiary and quaternary structures. Enzymes are biological catalysts or biocatalysts and they mostly speed up the rate of the biochemical reactions. Under optimal conditions, most of the enzymatic reaction can proceed at 10 to the power 8 to 10 to the power 11 times 10 to the power 11 times more than corresponding non-enzymatic reactions. How do they speed up biochemical reactions? Like all catalysts, chemical catalysts, enzymes work by reducing activation energy of chemical reactions. Now, what is activation energy? We can understand it through this figure. Activation energy is the minimum amount of extra energy required by a reacting molecule to get converted into product. In this case, this is the amount of activation energy. Now, watch figure 2 very carefully. Green curve is representing non-enzymatic reaction. In absence of enzyme, the activation is this amount, whereas in presence of enzyme, the activation energy is very less. As less activation energy is required for enzymatic reaction, so time required for substrate to product conversion is less. Now, what do you think? Without enzyme, biochemical reaction is possible or not? Yes, biochemical reaction is possible, but the speed of the biochemical reaction will be very less in absence of enzyme. Without enzymes, most of the reactions can't take place at a perceptible rate. Although enzymes accelerate biochemical reactions, they do not alter reaction equilibrium. They accelerate the forward and backward reaction by precisely the same factor. They accelerate the attainment of equilibrium but don't shift the equilibrium. Why? The equilibrium constant is related to delta G0 as K equilibrium is equal to e to the power minus delta G0 divided by RT, where delta G0 is equal to the change in free energy for the reaction. If you see this figure, you can see that delta G0 value, that is overall energy released during the reaction process, is same for the enzymatic reaction as well as the non-enzymatic reaction. See, this value is same for both of the enzymatic as well as the non-enzymatic reaction. So delta G0 is equal to the change in free energy for the reaction, which is equal, value of delta 0 is equal for both of the enzymatic as well as the non-enzymatic reactions. Therefore, you can see for non-enzymatic as well as enzymatic reaction, K equilibrium is same because delta G0 is same. Therefore, I told you that enzymes do not alter the reaction equilibrium because the K equilibrium is same for enzymatic as well as non-enzymatic reaction. Enzymes are highly substrate specific. One enzyme may bind with one or two substrate depending upon the particular chemical reaction. If A is a substrate and B is a product, imagine E enzyme is involved in this reaction. So in this process only one substrate is there, which is A. Now if A and B, A plus B, both are substrates, in that case enzyme E is required for this reaction and enzyme E can bind with both A and B. Then the product will be either P or it may be P1 plus P2. P2. Only a certain region of enzyme, which is called active site, binds to the substrate. See, this is an enzyme, model of enzyme. Here, this portion is active site. This is active site part. Substrate can only bind at active site, not here or here and there. The active site is a groove. This is a groove. This is a groove or pocket formed by the folding pattern of the protein. As you know, enzymes are proteins, I told already. This site is composed of unique combination of amino acid residues with different properties, which create a very specific chemical environment. Therefore, like a jigsaw puzzle, or like you can imagine it like a lock and key, a specific substrate matches this site and makes the enzyme specific to this substrate only. If the structure of the substrate is like this substrate can't bind at this active site of this enzyme. Now, say this two chemical reactions. If A is the substrate for E1, then E1 enzyme can't bind with substrate C. Only E2 enzyme can bind with substrate C, not with A. I hope it's clear that enzymes are highly substrate specific. In short, we can say that enzymes are mostly proteinaceous in nature except ribozyme, which is RNA. Enzymes are biocatalysts, which regulate rate of biochemical reactions, mainly speed up the biochemical reaction rate. They do not alter the reaction equilibrium, and they are very much substrate specific. Hence, we can define enzyme as it is an organic substance that acts as a catalyst in living organisms, regulating the rate of chemical reactions without itself being altered in the process. There are several factors that affect the speed of an enzymatic reaction, such as concentration of the substrate, concentration of enzyme, time, temperature, pH, and presence of activators. Effect of substrate concentration. When other factors like enzyme concentration, time, pH, temperature are constant, the rate of enzymatic reaction increases with the substrate concentration until a limiting rate is reached. After that, further increase in the substrate concentration produces no significant change in the reaction rate because enzyme molecules are completely saturated with the substrate. So this is the pattern of the enzyme activity versus substrate concentration graph. Similar pattern we can see when enzyme concentration is increased, but substrate concentration, time, pH, temperature are constant. Initially, as the concentration of the enzyme is increased, activity proportionately increases. As more enzymes are available to form enzyme substrate complexes, then it remains constant, or we can found a plateau, because there are not enough substrate molecules to bind with the extra enzymes. Enzyme activity increases with time, remains constant, and then drops when all other conditions are constant. With very long time of incubation period, enzyme denaturation is occurred, so activity decreases. Denaturation means enzyme deformation or enzyme structure deformation. At denatured state, enzyme can't work. As enzymes are protein, they are highly sensitive to thermal changes. Every enzyme works very good within a certain temperature range and most active at a specific temperature, which is called optimum temperature of that enzyme. Few enzymes are cold active, can work at very low temperature. Few enzymes are thermotolerant. They are very active at higher temperature. But few enzymes are also present which act at normal room temperature. We'll get this type of curve for each of the enzyme if you plot activity versus temperature graph. We found similar pattern in case of pH. Enzyme activity increases with change in pH till a certain pH, then decreases. Some enzymes are acid stable, can work at a lower pH range, whereas there are few active enzymes at higher pH range. Each enzyme is most active at their optimum pH. Effect of different activators on enzyme activity. Certain inorganic metallic cations like calcium, magnesium, manganese, zinc, cobalt, copper, sodium, potassium, iron, etc. activate enzyme. Hence, in presence of one of those metal ions, enzyme activity increases. Very few anions act as enzyme activator. As for example, chloride ion is required for amide.
I hope you understood the topic very well. Don't forget to like, share and subscribe this channel. I'm going to upload various videos on enzyme regularly. Therefore, press the bell icon for further notification. Take care. Bye bye.